Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, today we are going to start with chapter number 8, Cell Biology and Biotechnology. So in the last lecture we had finished with chapter number 7, and today we are going to start with chapter number 8. So let us start with cytology. Earlier we have studied the structure, types and organelles of the cell. This is called as cell biology or cytology. So cytology is the study of cell and its part. Besides above mentioned points, it includes the study of cell division and many other aspects of cell. There are revolutionary changes in the field of human health due to cell biology. Research institutes specially dedicated for research on cells are established at Pune and Bengaluru in India. National Center for Cell Science at Pune and INSTEM at Bengaluru are involved in valuable research. So these are the institutes in our country developed for cell research. Now let us talk about stem cells. These are special type of cells present in the body of multicellular organisms. These cells give rise to all other types of cells present in the body of multicellular organisms. Similarly, these cells play an important role in wound healing. So stem cells are the special type of cells present in our body, and this cell give rise to all other type of cells. And these cells plays an important role in wound healing. We had studied the stem cells of plants in previous class. Now we shall study the stem cells in animals, particularly in human beings. New organism is formed from the zygote that is formed by union of male and female gamete. At the earliest stage of development, organism is in the form of mass of cells. All the cells in that mass are almost alike. Those cells are called stem cells. So, during the formation of zygote, then development of zygote, there is a group of cell called mass of cells, and this mass of cell are nothing but stem cells. Now this part is being removed. Starting from here to here, this part is eliminated. So let us talk about stem cell research. In biotechnology, stem cell research is a revolutionary event for after cloning. This technique has the potential of bringing about the fundamental changes in the medical science. Depending upon source, stem cells are of two types, as embryonic stem cells and adult stem cells. So stem cell research is one of the most important technique developed after cloning. And upon the source, stem cells are of two types, embryonic stem cells and adult stem cells. So what is embryonic stem cells? Division of the zygote starts and thereby it is converted into embryo. Cells of embryo undergoes repeated mitotic division. Cell differentiation starts from the 14th day of conception. Cells of different organs like osteocytes, that is bone cells, hepatocytes, that is liver cells, and neurons are formed due to differentiation. Embryonic cells before the differentiation are called embryonic stem cells. 220 different types of cells in human body are formed from single type of cells, that is embryonic stem cells. Thus, stem cells are primary type of undifferentiated cells with self-multiplying ability, and they are parent cells of all types of human cells. This property of stem cell is called as pluripotency. It has been found that if these stem cells are collected well before the beginning of differentiation on 14th day, that is during 5th to 7th day, and cultured with certain biochemical stimulus in laboratory, as per the stimulus they can transform themselves into desired type of cells, thereby tissues, and finally into organs. So embryonic stem cells are the stem cells formed during the formation of embryo. And these embryonic cells have this ability that they can form tissues and finally organs if they are cultured in proper medium. Now let us talk about adult stem cells. Stem cells can be obtained from the body of adult person too. There are three main sources of stem cells in the body of adult persons. Stem cells can be obtained from red bone marrow, adipose connective tissue, and blood. Besides, stem cells can be obtained from cord blood immediate after birth. So, immediate after the birth of baby, the cord attached to the mother womb, even that contain stem cells. Now, what are the uses of stem cells? Very important. Regenerative therapy. Stem cells are used to replace the dead cells in case of conditions like diabetes, myocardial infarction, Alzheimer's disease, Parkinson's disease. To produce blood cells required in conditions like anemia, thalassemia, leukemia, etc. So here where cell therapy is carried out with the help of stem cells. Now if you talk about organ transplantation, in case of failure of organs like kidney and liver, those can be produced with the help of stem cells and transplanted. So even for organ transplantation, this can be used. Organ transplantation. Various organs in the human body either become less efficient or completely functionless due to various reasons like aging, accidents, infection, disorder, etc. Life of such person becomes difficult or even fatality may occur under such conditions. However, if a person gets the necessary organ under such conditions, its life can be saved. Availability of donor is an important requirement in organ transplantation. Each person has a pair of kidneys. As the process of excretion can occur with the help of single kidney, person can donate another one. Similarly, skin from certain parts of the body can also be donated. Various factors like blood group, disease, disorders, age, etc. of the donor and recipient need to be paid attention during transplantation. However, other organs cannot be donated during lifetime. Organs like liver, heart, eyes can be donated after death only. This has led to the emergence of concepts like posthumous, that is, after death, donation of body and organs. Organ and body donation. Human bodies are disposed of after death as per traditional customs. However, due to progress in science, it has been realized that many organs remain functional for certain period even after death occurs under specific conditions. Concepts like organ donation and body donation have emerged recently after realization that such organs can be used to save the life of other needful persons. A liberal view behind the concept of organ and body donation is that after that, our body should be useful to the other needful person so that their miserable life would become comfortable. Awareness about these concepts is increasing in our country, and people are voluntarily donating their bodies. Life of many people can be saved by organ and body donation. Blinds can regain the vision. Life of many people can be rendered comfortable by donation of organs like liver, kidneys, heart walls, skin, etc. Similarly, body can be made available for research in medical studies. Many government and social organizations are working towards increasing the awareness about body donation. So, students, body donation is a very important aspect one can think of. In this body donation, one can make a will that he or she would like to donate their body parts after their death, and that body part can be transplanted into the another body person who is in need of it. So this way the person's life can be saved even after the death. Now let us talk about organs that can be donated. So you can see here the organs like lungs, liver, bone marrow, its pancreas, heart, eye, kidney, skin, all these organs can be donated. Now let us talk about biotechnology. We have studied in the earlier class that biotechnology is bringing about artificial genetic changes and hybridization in organisms for human welfare. Various branches of science like cytology, biochemistry, molecular biology, and genetic engineering are included in biotechnology. There is considerable progress mainly in the field of agriculture and pharmacy due to biotechnology. New experiments are being performed for improving the agricultural yield. In pharmacy, experiments for production of antibodies, vitamins, and hormones like insulin have been successful. High-class varieties of crops have been developed through the technique of tissue culture. So in the ninth standard, we had a chapter Introduction to Biotechnology, and in that we had studied about how biotechnology is nowadays in great demand and helpful to us. Biotechnology includes following main areas: use of various abilities of microbes like yogurt production from milk and alcohol from molasses. Use of productivity of the cells, example production of antibiotics and vaccine with the help of specific cells. Use of biomolecules like DNA and protein in human welfare. Development of plants, animals, and production of desired quality by gene manipulation. Production of human growth hormone with the help of genetically modified bacteria. Use of genetic and non-genetic technique. Non-genetic biotechnology involves use of either cell or tissue. Example, tissue culture, production of hybrid seeds, etc. So these are the areas where biotechnology is used. Now are the benefits of biotechnology. It has become possible to increase per hectare yield irrespective of the limitations of cropland. Expenses and disease control have minimized since development of resistant varieties. Due to development of fast fruit setting varieties, yield per annum has been increased. Development of stress resistant varieties which can withstand variable temperature, water stress, changing fertility of soil, etc. has become possible. So these are the benefits of biotechnology. Now what are the commercial application of biotechnology? Crop biotechnology. Biotechnology is used in agricultural field to improve yield and variety. Hybrid seeds, gene of two different crops are recombined to form hybrid of various crops. This is especially used for fruits. Genetically modified crops, Crops which develop with desired characters by integrating foreign gene with their genome are called GM or genetically modified crops. High yielding varieties with resistance to diseases, alkalinity, weeds and other stresses like cold and drought. Bt cotton is one of the examples. A gene had been isolated from the bacterium Bacillus thuringiensis and integrated with the gene of cotton. Due to this, the toxin, which is fatal for bollworm, was produced in the leaves and bolls of cotton. The bollworm feeds on leaves. The toxin destroys its alimentary canal and the bollworm dies. Bt brinjal. Bt brinjal variety is developed by using the gene isolated from the Bacillus thuringiensis. This improved variety of brinjal kills the pest in same way as the Bt cotton does. So this Bt cotton and Bt brinjal are the genetically modified crops. Let's talk about golden rice. A gene synthesizing the vitamin A, that is beta carotene, has been introduced in this variety of rice. As compared to the normal variety, this variety, which has been developed in 2005, contains 23 times more amount of beta carotene. Herbicide tolerant plants. Weeds always affect the growth of main crop. What are weeds? These are unwanted plants that grows along with the crop. If herbicides are used to destroy the weeds, it affects the main crop too. Due to this, herbicide-tolerant plants, varieties of crops are being developed. Due to this, it has become possible to selectively destroy the weeds. Biofertilizers. Due to use of biofertilizer instead of chemical fertilizer, nitrogen fixation and phosphate solubilization, abilities of the plants are improved. Mainly, the bacteria like Rhizobium, Azotobacter, Nostoc, Anabaena and plants like Azolla are used as biofertilizers. In the last year, we have studied the tissue culture. Genetic improvement of the plants has become possible due to tissue culture and besides those characters inherited in the next generation. So biofertilizers are the biofertilizer which are used instead of chemical fertilizer. And here the bacteria like Rhizobium, Azotobacter, Nostoc, Anabaena and plants like Azolla are used as biofertilizers. So students, we'll stop here. Now in the next lecture, we'll discuss the further topic of this chapter. By the time read this part, Assalamu alaikum wa rahmatullahi wa barakatuh.